Thank you for the introduction, and thank organizers for giving me an opportunity to talk in this kind of a forum, because as you can see from the title and also from the background picture that you see here, that you would not see any equations or whatsoever, except maybe for one slide in this case. But it is still interesting. I thought that these proteins are something that, or the cytoskeleton has been something of interest to the theoreticians and physicists for a long time. And of course, the entire biology is run by this physical phenomenon. So the title of my talk is Understanding the Basis of Diversity in Cytoskeletal Organization, PROKARYA. And I think I need to put this PROKARYA in quotes. And most of this work that I'm going to talk about has been actually done in Mohan Balasubramanian's lab, where I was a postdoc then, at Temasek Life Sciences Laboratory in Mechanobiology Institute, and Mohan himself has moved to University of Warwick recently. So, of course, why PROKARYA? They are the minimalistic of any complex systems that you can see, and it's always easy to study them and model them, as we see that simple systems are more easy to work with. And in fact, it was to a level that when the entire concept of a bacterium was being defined, identified, Roger Stanier and von Neil at that time in 1962 went on to state that there are only two kinds of organization that you can see in life forms. One is this beautiful pictures that you see around here for the eukaryotic family, where you have microtubules, actin, and intermediary filaments doing lots of work and motors running on them, which will carry a lot of things and do a lot of functions in them. And the other organization that life had was basically this. This is in prokaryotes, believed to be at that time that there was a organization whatsoever within them. And in fact, it was as simple that they just grow as a soap bubble grows as you pull them, and it will also divide just as a soap bubble but snap. So it was as simple as such. So even then, there was interest from physicists in these kind of models, but it was thought to be extremely simplistic like this. However, over the last 20 years or 20 years, because all this view changed, with the identification of proteins in bacteria that look like the classical cytoskeletal proteins that we know in eukaryotes, as the tubulin and actin-like proteins. And further, it was seen that they also localize in bacteria to certain locations and do functions just like the eukaryotic counterparts do. And in fact, today we know that the eukaryotic counterparts are actually evolved from the prokaryotic homologs. And every single one of them has an homolog in the bacteria, deciding the cell shape, deciding whether the cells are rod-shaped or how the plasmids are segregated and involved in cell division, and a unique class of cytoskeletal proteins in bacteria which are called simple Walker AATPases. So these are just ATP hydrolyzing molecules. And in fact, they are organized within the cell. So this is one of the actins that seem to be around spirals, but now we don't think it is spirals. And there are proteins that look like microtubules that pull the plasmids apart and push the plasmids apart. And here is the cell division organizing ring that is a contractile ring without any motors. It's contractile without any motors. And today we know nearly more than 50 different families of these proteins. So that is the history to this entire cytoskeletal protein. But while so many different families are known, in the eukaryotic world we know that the actin-like proteins has a property called as treadmilling, which is always attributed to it, wherein it simply means that it is growing at one end and kind of addition at the other end is slower than the one end. So it seems like it is growing from one end. And another famous property that is known as dynamic instability, where the polymer is simply growing and then shrinking. It just grows and shrinking. And these kind of things can be easily visualized in eukaryotic cells by simply fusing them to fluorophores in biology. So here's an actin that reorganizes itself into a ring-like structure. You can see the cables being pulled on and being organized into ring-like structures here. And here is a microtubule that is growing and shrinking. So this one will shrink. Sorry? Yeah, so these molecules, this one would actually be treadmilling. So these cables would actually be growing at one end in a treadmilling. And this is the dynamic instability seen in microtubules. They just grow and then shrink. So, of course, these were not known for the prokaryotic homologs. And nearly 10 years ago, when I started off with the postdoc, what we simply did is to take these prokaryotic homologs and put them in an eukaryotic environment just to see what the polymer by in itself will do. Studying in the bacteria would always complicate it with other negative regulators and positive regulators, which would influence its dynamics, which is of course essential to study. But we thought we would take a very simplistic approach and see what the polymer themselves would do like in a heterologous environment like this. And it was very easy to imagine POMBE. It has this beautiful cylindrical shape which can be put on agar slides, and we were able to image. And this kind of gives rise to a lot of interesting things what we could do. We could not do with prokaryotic cells, we could do with this. One of the most important thing is this. These proteins, the cytoskeletal proteins of bacteria, are essential. So any tinkering with them would cause the cells to change or not even allow them to grow. So in case of fission, we were able to make umpteen number of mutants that would not affect the cells and still study them. So in that process, we had established fissionist as a system to study the prokaryotic cytoskeletal systems for a large number of proteins. In fact, all the proteins that were there. And in fact, we also identified a new cytoskeletal element from Archaea, which is the escort-3 family, using the system. And to summarize the entire thing, which I'll now talk about a bit more in detail, this is an actin-like protein expressed in fissionist. This forms these long polymers. And when you alter the cell shape here, the polymers are no more oriented longitudinally, and they are randomly oriented in the spherical cell. So again, this geometry decides what the cytoskeletal organization inside the cell could be. And here is the case of a tubulin of the FTSC that forms rings in bacteria. Here as well, we can see that it forms these tiny rings, and we could show that these rings are formed by a spooling process. And there's another tubulin-like protein that serendipitously went into the nucleus of the yeast and is able to push the nuclear membrane and deform the nuclear membrane. And I'll talk about some of these work in the next few slides. So in the current lab, what we are currently interested in, two very simple aspects in bacteria of life. One is you need to partition its DNA for the life to survive. The daughter cells should receive equal number of its genetic material. And secondly, such partitioned genetic material should be also cytoplasmically partitioned, and they should be separated. That's the cytoplasmic partitioning. That is the cell division process. So these are the two processes that my lab would be interested in at NICER. So coming to cytokinesis, as I told you, in eukaryote, it is the actin that forms the ring. So it's an actin-like protein, but in bacteria, it is the tubulin homologue, which forms this cytokinetic ring and is actually contractile. And what we found when we expressed in yeast cells that they do not go to the ring in yeast in this case, it's completely heterologous system. It also self-organizes into these ring-like structures. So that was interesting. You don't need any bacterial accessory protein to actually take this protein tubulin homologue into a ring-like structure. So it can just spontaneously form into ring-like structures in any cell, any system. Drives this, so that is something which we don't understand, but I'm just coming to that, probably what it could be. So we made a number of mutants to understand how this ring formation actually happens. We made a number of mutants, and as expected, this is a protein that binds GTP, and that drives the polymerization. So if you do not have any GTP binding, you don't have any polymers formed. And if you reduce the GTP hydrolysis rate, they don't form rings. So probably it is GTP hydrolysis that might be driving the ring formation. But this also has reduced GTP binding. So one of the most interesting mutants was this, which can bind GTP as normal to this wild type. However, it has a reduced GTP hydrolysis. So it forms these long filaments, and there is another population of cells which has single rings, and some which look like connected here. Not rings, yeah. So, but the thing is that also it binds some GTP, but it is also reduced in GTP hydrolysis. So it may be both linked there. So when we started imaging these cells which have these long filaments, this is what we see. So you can see at some point it curved, and the entire filament is being drawn into that, and it's kind of spooling. So the way, and you see at both the ends because the cell just divided here and it just broke the filament. It's just a physical breakage of the filament. So therefore, then you see that this one is swirling on here and going on to form the ring, and this at the other end. But otherwise, it has nothing to do with the cell cycle or any other process in the yeast. So we could do this even with multiple rings and this one and observe the same thing. So for what we did is, if this is the protofilament axis, yeah, so the filament just broke. So even otherwise, if there were shorter filaments, it would still go on to form randomly some ring-like structure. So it has nothing to do with the filament breakage or anything. So we don't know what exactly triggered the ring formation in this case. So in this case, it was breakage somehow correlated with breakage. But there are other cases where there are multiple filaments, and each of that without any breaking event, they would just go on to form the ring-like structures. That's the model that we think, and that is what we think, but it may not be completely true with some recent data that is coming up. So I'll just touch upon that at the later stage. So what we did at that time is, then if this is the protofilament for the polymerization, this is one subunit, and the next subunit will sit on top of this, and therefore so on and forth, with GTP binding somewhere here. So this was the mutant I showed you, the point here where the GTP hydrolysis is not there, and it forms this tight ring-like structures. But when we added another mutation on this surface, assuming that the protofilaments, one protofilament will be sitting here, another one sitting here, to form that ring-like structures, they never form the rings. So they are just linear filaments. This never curves, this never forms the rings. So then the idea was this, taking analogy from the microtubules, that monomers upon GTP binding would form these linear filaments, and GTP hydrolysis would trigger the curvature. That would lead to the ring formation. And in fact, the scenario would be something more like this, because FTSZ is known to be short filaments or 150 nanometers in vitro conditions. So then this will be multiple filaments attached to each other and then contributing to the ring formation. But of course, this was just an take. So in microtubules, it does to curvature, because there is this, initially the filament is very straight with GTP binding, and upon GTP hydrolysis, I think the filament gets some kind of, it bends by around 15 degrees or 30 degrees here. But in this case, we don't, GTP hydrolysis leads to a much larger curvature in this case. So why it leads to curvature, that is something which I don't know how to answer this question actually. So what we could say at least at that time, that the FTSE ring assembly is something of more spontaneous in nature. It self-assembles into your rings, does not need any bacterial accessory proteins. It's an intrinsic property to this protein, and it forms like a spooling-like process. But however, we also took a look at these ring-like structures by EM. So how many filaments are there in this is something which we wanted to see. So what you see by GFP fluorescence is not a single protofilament of that 4 nanometer protein. So it seemed like multiple subunits. So what you see here in each of these black lines is the protofilament, which is 4 nanometers, and the entire ring consists of multiple protofilaments arranged laterally to each other. So this may be more clear. So this is just one section of that entire ring in EM. So each of this is a protofilament, and it's not that these are concentric rings, but this is just one section. So this might go around and go into the next filament in the next section. So these are not concentric rings here. So we try to trace each of these filaments, and it looks something like this, but there's not a real tracing of the filaments. So we could not trace the filament in continuity. So it was not very high resolution image, or we could not completely trace it. It was a bit complex to do that job. Yes. Yes, this is still in the yeast system. I have not gone to the bacterial system. So I'll just tell you what happens in bacterial system. So we had just modeled this crudely with sorry, very sorry. And it seems like this is the compact ring, that entire ring structure, not the wild type, but the mutant with GTP hydrolysis that forms this entire, all the filaments are going into a single ring-like structure. So what happens in bacteria, we never saw the wild type. No, it doesn't seem to be. It seems like it is there as some length, and then it's attached by, it's like something like not a continuous cable. So you have one cable, and then you have another filament that is somewhat attached to this filament, and then it is a continuous thing. It's a multiple filaments that are sitting together. So it's not one single long, very long filament. In fact, the entire diameter of this could be nearly 400 nanometers. Inner diameter is 200 nanometers. And there is this height is around 600 nanometers, so it's almost like a barrel there. So that is we don't know exactly how they bind, but that is the idea that I told you here, that this might be multiple filaments that are just laterally associated here. So what are those residues, we don't know, but one of those residues might be this. So if this is the protofilament axis, this will be the lateral interaction axis, and these residues might be involved in stabilizing the ring, as this mutant does not go on to form rings. Any other accessory proteins, yes. So they would just bundle, but this requires them to be in a molecularly crowded environment. So that is the cytoplasm of the yeast. So that is why we were able to do this in yeast, and in vitro, it will be possible to do it when you add molecular crowding agents. So that has been done by others. Or when it comes to the membrane. So if you add an artificial membrane tethered to the FTSE, then the membrane can actually bring it together and create this molecular crowding equivalent. Okay, so this is what happens in the wild type scenario. This is a mutant. So we never could image the wild type in the yeast cells at that point of time, because probably it was just few filaments and it was lost in the contrast in the yeast cells, which is highly contrast. The cytoplasm is also having the same contrast. But in vitro and in vivo, Ian Lowe's lab did cryo-electron microscopy. These are not cryo-electron, but they did cryo-electron microscopy. And in dividing E. coli cells, in bacterial cells, and in vitro reconstituted systems, they could see that the filaments are something like this. So this is what, so these interact with some distance here, but we don't know the nature of those interactions, but they can be associated laterally here. So this is one filament, there's another filament here, and eventually they constitute the ring in the bacterial cell. So these are short filaments that organize into a ring-like structure in bacterial cells, and that somehow leads to a constriction and sliding. So the hypothesis that they have is it's a sliding filaments that actually lead to constriction. But how do filaments slide? What leads to sliding? Because there are no motors in this case. Unlike in the actomyosin ring, where you have the actin ring and myosin associated with them and there is a sliding phenomenon, we don't have any motors in bacteria. No. So the yeast system is completely, it lacks any of the bacterial homologs or anything. They are happy just forming rings by themselves. Yeah. And that's why there is not any localization or anything like that. Multiple rings. So in the case of wild type, it is multiple rings. So if I multiple rings, exactly. So the single ring is only in this GTP hydrolysis mutant. So it's allowing, what it is simply doing is hydrolysis leads to depolymerization. So what the hydrolysis mutant is allowing you to do is it's allowing you to polymerize up for a longer time, because you don't have any hydrolysis. So you form the single long filament and then a single ring. Everything comes as one polymer and then forms the ring. But in case of wild type, where the hydrolysis rates are faster, you have depolymerization, so you have very short filaments that will go on to form rings. So it might just be 3 filaments from this entire structure. That will be the wild type structure. And in fact, that is what is seen in this E. coli, that these rings are just 3 or 4 filaments in width, not more than that. Here I have absolutely no clue how many filaments are there, because this entire thing is nearly 600 nanometers. It's almost like a barrel. So it's too much of protein there. We took in our lab, and what we're doing currently is, if this entire thing is a ring and this is being compacted into a ring-like structure, we want to ask the question like, what are the molecular nature, or what leads to this ring formation? So if it is compacting in this way, so we identified mutants, we just randomly mutagenous as proteins, and identified mutants that actually gave rise to this structure. So I showed you a compact ring, and these are helical structures here. So there were 2 mutants that were identified with different helical pitches. We need to get better imaging of this done. But what it seems like is, if we had to take this ring and hold at the 2 ends and just pull them apart, this is what this structure would be. So these are somehow defective in actually compacting into a ring-like structure. And that can come from 2 kinds of things. One is the contacts here are being lost and they are just loosened up. Or these individual protofilaments in this case probably are more stiffer that they cannot really get that compression into a spring. So if you had a very thick spring, you would not be able to compress easily. So if the filament stiffness has gone up in these mutants, that would be something. So the previous one is just a hydrolysis mutant. So this one, we don't know what the nature of the mutation. We just got this, we just mutagenized everywhere. And 2 of these mutations led to this kind of structures. So radius will be, we presume this will be equal to 250 nanometers in this case. We don't know. We again tried EM for these helices, but we could not see anything. So the only structure that we could see by EM was this. We could not see any of these structures by EM. So this is something that we are continuing. We are taking these back in bacteria and see what their function might be as a cell biological function in bacterial cells. So one of the things that we can at least say is that the helical-like structures is compacted into ring-like structures, which has been seen in bacterial imaging as well. In some cases where before the cell division happens, FTZ is present like helices attached to the membrane. But as the cell division progresses, it forms this compact ring-like structure. And what we are interested in to see is how these are modulated by various other inputs from external cues. And of course, this gains significance because a lot of bacterial pathogens block cell division when they are inside the host. So these are salmonella cells or Mycobacterium tuberculosis that just form filaments and they do not undergo division when they are inside the host cells. And this way they probably evade eating by the macrophages when they are outside. Example, in the case of urinary tract infection, macrophages cannot eat simply because the cells are longer. And in other cases, they just evade the immune system. So just switching to the second part of the talk, the DNA segregation systems and bacteria. So in most simplistic form, DNA segregation requires just these elements. So you have a piece of DNA, and there is a protein that recognizes an element on the DNA. These together are called as a kinetochore in case of the eukaryotes. And there is some kind of polymer that would recognize this complex and then push them apart or separate them apart. So that's the essential components that would be needed. And that is, entire thing is extremely complex in eukaryotic cells where the kinetochore itself contains around hundreds of different proteins. But in bacteria, you can get to the minimal here. This is just in one element. This is a single protein. This is a single protein that forms the polymer. So you have just 3 components doing the job. So it cannot get more minimalistic than this. And one of the most well-studied protein is this PAR-MRC complex, where the kinetochore or the PAR-R binds the DNA here. And the polymerizing protein, when it is bound to a single plasmid, it keeps growing and shrinking. So this is the dynamic instability that I talked about for microtubules, but this is an actin, but still does the same thing. But as soon as you have 2 plasmids, that is the replicated plasmids, the end gets stabilized, and through an insertional polymerization at this end, the filaments just grow. And as they grow, they push the plasmid apart, and the cell divides in the middle, ensuring that the plasmids are transferred to both the daughter cells. So this you can just. So as soon as there are 2 plasmids, this gets stabilized, and then there is polymerization, there is no depolymerization, and this keeps pushing the plasmids to the 2 ends. And the cell would just divide right here. Yeah. So even chromosome, we know in case of Caulobacter crescentus, one species at least, that this is the same thing that happens. Similar thing happens. Yeah. No, not we don't think so in this case. But it seems like there will be some. So of course, how plasmids are segregated has been studied for a long time, and there have been several models. One has been the simplistic old model where the chromosomal DNA just binds to the plasma membrane, and as the cell elongates, the DNA would be pulled along as the cell is elongating, simply because of cell wall synthesis and elongation of the cell. There have been other models where gradients have been made in the PAR-MRC complex, and the DNA or this DNA-bound protein complex is being pulled by this gradient. And there are these filament models where the complex is bound here, and like a mastic is burning, it just burns and drives the hydrolysis, and the plasmid, the DNA complex is being pulled to the other end. And more recently, there have been like models which random diffusion model, and other model is where the DNA itself is having some elasticity and that influences the movement. So all of these models are essentially the same, where there is some kind of gradient of the ATP bound form and the ADP bound form. ADP bound form releases and ATP bound form binds to the protein, and there is a gradient of ATP to ADP bound form in this case. So although they have some commonality in the system, they have some kind of differences in the details. So one thing that I would talk to you about is this filament forming proteins. So I told you about an actin-like protein which is well studied, but there's another tubulin-like protein that was recently identified from a species called Bacillus thuringiensis, and we were able to show that that has a growing end and a shrinking end. So this is that in time. So you have this end is actually depolymerizing, or it is shrinking, and this end seems to be growing. This is in the yeast system, and it was also shown in the bacterial system by the same group in this paper, and later we could show in the yeast system. This TUBZ. So, and there is also recently identified nearly 40 different family of actin-like proteins from bacteria. And most of these bacteria are not even culturable. You cannot study them, you cannot put them plasmids, you cannot genetically do anything with them. So, this one, yes. this would be induced by the surface. So if you had a round cells, it might just be more curved. So what we did is one of these proteins that we took from TUBZ from Bacillus thuringiensis, we expressed it, and serendipitously it went into the nuclear. So this is the nuclear envelope. It went into the nucleus simply because it has some sequences that will take it into the nucleus. And inside the nucleus, when it polymerized, it was able to deform the membranes here. In fact, this is what will happen with a normal yeast cell if we did not have this molecule. So this is a yeast cell which is having the microtubule. So this is a bacterial tubulin, but this is the yeast tubulin, which will be anchored to two of these spots that will make it rigid, and that lead to the separation of the nucleus. So this is the spindle in yeast. If you did not have this attaching complex here, what it would lead to is this microtubules inside would just push the membrane and they would deform the membrane. And what we could see is this kind of similar phenotype was observed with these bacterial proteins pushing the membrane. So we utilized this membrane shape in this case and bending of this one together with collaboration with Keng Wee at IHPC in Singapore. What they did is they measured these yeast nuclear envelopes, the deformation in the nuclear envelope, and also the deformation in the filaments that I showed you. And this is the only slide where you will actually see an equation. And they could calculate a force generation by these bundles on the membrane to be around 1 to 10 nanonewtons, and which is very similar to the ones that is actually made by microtubule filaments. Okay, so this is something that we could further be interested to actually see, do a better modeling, actually take more of the nuclear images and see how much force we can actually get from these microtubule-like proteins and measure more quantitatively how we are able to do this. And this apart, I just told you about different actin families from different bacteria where we cannot even culture the bacteria. We utilize our yeast system to actually express each one of them in yeast, and they all form polymers. So this is something which we did not expect because it was just based on ATP hydrolysis motif that they had predicted. But all of them form polymers, and each of them have kind of different polymer morphologies in this case. And we picked some of them, and electron microscopy in collaboration with David Pop here in Singapore, they did the EMs and electron crystallography, and we started to see varying morphologies. Yes, it is in the nucleus. So that is, it will be kind of a confinement in this case. This is a yeast cell. So this is again, you are just confining them to the yeast. So this will be the boundary of the cell. Cylindrical, yes, these are cylindrical cells. The yeast cells are cylindrical cells by themselves. Yeah, so because yeast cells grow, fission yeast grows as cylindrical cells, that is how it comes from. This is around 7 microns. This will be around 7 microns. Some of them are even longer. Like this one, you can see, width will be 4 microns, 4 to 5 microns. In fact, in this case, the polymer you can see very thick, but we can see that the polymer is still sticking on there. It has not, the septa has been able to cut the polymer in this case. So what we I showed you an actin that is just a filament like this. But what we started to see with some of these proteins is that you can start to get different helicities and different orientations. What you see here is one actin filament, double-stranded, that is going this way, and the other is an antiparallel. So this filament would be going away from you, and this would be the filament that is actually coming towards you. So this kind of actin formed an antiparallel filament, which was not seen in earlier any form of actin. So this is something that was new to the actin cytoskeleton filaments in bacteria. And yet another protein, again from Bacillus thuringiensis, which an actin, formed these very straight, rigid rods in yeast cells. Again, this is a cylindrical confinement, like in this is this one cell that you can see here. So this is filaments. And what we could show with this protein is that this is in fact a bona fide from bacteria, expressed in yeast, yes. In the bacteria, we don't know. But that one was an in vitro. So we completely purify the protein. So again, we don't know the structure in the yeast. It's a purified protein without any other factors or anything, just purified protein EMs. In this case, we were able to show that this is actually a bona fide plasmid segregation protein. that this is the polymerizing protein. This is the DNA binding protein that I talked to you about. So this protein, by some experiments like gel filtration or DNA binding experiments, we can show that this protein actually binds to the DNA element here. So this is one case here, this protein actually binds here, and therefore the mobility is slower. So as we increase the concentration, you get more and more binding here. And also this particular protein interacts with this protein as well, because both are coming down together in a sedimentation assay here. And here again, purified protein in vitro has a very different actin-like, although the monomer is like actin, the filament is very different from that of an actin. It's just turning around with different helicities here. So this is these are two parallel filaments. So one filament is going here and the other filament is going the other way. And in fact, the addition of this PAR, so in vitro, this protein, it changes the actin structure completely in vitro. So here, these are just two filaments. One is minus and plus. The other is again, this is the minus here and this is the plus here. For this is the minus. So these two are anti-parallel in themselves, unlike the earlier one where the two were actually parallel filaments. And then there was an anti-parallel filament. In this case, this two single, and then these two anti-parallel come together to form yet another anti-parallel. So you have plus minus 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 plus. And it forms a tube-like structure when you add this protein para to it. So it really changes the structure. So you can have additional factors that might change the structure. So this is a summary slide where you can see this. These are the microtubules, which are like 25 nanometers in tubes. But this is a tubule formed by this actin-like protein that I showed you now. So the tube is not very high, but what it suggests is that the bacterial homologs have tried out barriers during the course of evolution. The tubes were tried tubes made by these bacterial proteins. And this is an actin filament, and this without the PAR, this another TUBZ protein that I showed you. That is a minus filament. So you have a lot of diversity that is actually coming from its structural or the sequence diversity or structural diversity in the bacterial cytoskeletal proteins. And what this means and how they are functionally they are varying is something that we need to still see. So this is to show that the actin-like structure that I showed you is actually like behaving like a microtubule where it is shrinking and growing. So this was a treadmilling where there's a growing end and depolymerizing end. This one is an actin-like protein that is actually undergoing the catastrophe. So you reduce the energy, all of the protein completely depolymerizes. This place again you can see. So there's just completely depolymerizing. We just reduced ATP levels in this case, but the cell would have lost all its ATP, GTP, and all the energy components would go down. And this entire shrinking is actually bidirectional, as you can see by this bleach mark here. That it's not looping. Oh. So it's actually shrinking from both the ends, unlike microtubules, which is just growing and shrinking from one end. And this. So again, you can see the same thing here, that the polymer is actually shrinking, and then some of them are actually rescued. So as the energy levels come back, so they can start growing from the same end, just like the microtubule. So you just shrink and grow, start growing again. And unlike the microtubules, these again have the same property of the filaments coming together and sticking to each other. So this happens in a lot of bacterial proteins. We don't know why bacterial proteins tend to stick together and bundle like this a lot. And this is the structure in EM in the yeast cell. So this is one that we can see in the yeast cell itself, the EM structure. It has this interesting structure like going like a helical tube, but I'm not sure if this is a tube or just filaments sticking together laterally and they are just a sheet. So we have not done 3D reconstruction here. So it is just a single section. So it's possible that these are just the filaments that are orienting themselves and bundling laterally here and forming a sheet-like structure. It's also possible that these are structures, helices that are going around like helical structure, and it's actually a tube-like structure. That is something which will be interesting. But again, what we would like to do is to take it in a bacterial system and see what they are actually in bacteria, rather than now going behind yeast cells and looking at what they form in yeast cells. But nonetheless, it is interesting because then it shows that varying environments can lead to different kind of polymer morphologies with the same protein. So this is again another example where an actin-like protein is just treadmilling in this case. So you have a shrinking end and a growing end. So this is the growing end, and this is protein when upon ATP, it just starts to depolymerize. So this is a steady state kind of behavior where ATP levels are normal in the cell and they are constantly there, so it kind of treadmills, and when you deplete them, it kind of goes into depolymerization. So again, what we have found with this is it doesn't matter what structure you have, but what matters is what kind of, how the polymers are added to the end or how the polymers are removed from the end. And that leads to this dynamic behaviors like treadmilling and dynamic instability. And actin-like proteins can do treadmilling, they can also do dynamic instability. There's a tubulin that treadmills, and recently it has been shown that FTS, the cell division protein, the ring protein that I showed you, even that also can treadmill. Although it's a single protofilament, it can do a treadmilling. And all of this work would not be possible without Mohan's lab support and a group at EMBL where all the EM work was done. So Claude Anthony helped us with a lot of tomography and EM work, and other collaborators with Madan Rao and Keng Hui and David Pope for the EM, and funding from Singapore Mechanobiology Institute, and currently from DAE and NICE. And thank you very much, and this is the nicer building. This is my son, so welcoming it. Thank you. Yes, exactly. So actins bundle, they bundle, but they need accessory proteins like actinin and several other proteins are needed in eukaryotes to actually bring the actin together to bundle them and keep them together. But in prokaryotes, you don't have such accessory proteins. It seems like it's built into the protein sequence itself. So you have sequences or amino acids at the lateral sides that will bring them together and make them stick in a particular way. But actins, eukaryotic homologs have evolved other proteins to actually bring them together when they need them to be brought together. So there's probably a next step of regulation in the course of evolution in eukaryotes.
That very nice sort of very ordered picture that you showed towards the end. One way you can get the sort of very ordered bundling is just through charge effects.
Yes, most
charge polymers. In some regimes, they tend to form bit much crystalline structures, which are.
Yes.
It depends very much on the nature of the counter ions.
Yes.
The charges on
yeah. So in fact, that might be true in these cases for all of these proteins that I showed you, FTSZ to actins and everything, because the cytoplasm is actually having a lot of potassium apparently, and if you change some residues, they also bind some magnesium or calcium. So if you change these counter ions, then the polymer architecture really changes. So it may be true for all of these proteins that in a very simplistic way, it is these ions that are actually influencing the polymer architecture here. But given that genetically to study, that becomes extremely difficult for us. Then.
It would be nice if it could be done in vitro.
In vitro, yeah.
Is it possible?
It is possible today. It's very much possible. To FTSE, a lot of this has been done by EM by changing the ions in vitro and looking at different morphologies. So the other actins are only recently people are started to work on it. So we know that it forms polymers only now. There are few groups they are working on it, but they have not changed much of this content. So we'll at least pick some of them and we'll do them in vitro. Yes. So another thing, again, more interesting thing is how those different helicities or different things come up for the same fold of the protein. So that might also be influenced by maybe by changing counter ions, you can change the helicity itself in these cases. No, we don't know the interface. Again, the EMs were not so good to actually find the interface residues that might actually be binding them together. So that again might be intrinsic to the filament. As they come, some of them will bundle parallelly. Some of them, the amino acids are such that they can bundle only when they are in the opposite orientation. I think so, because I showed you one image, when they are starting to form, they are probably just these two filaments, which might be more flexible and they may have the curvature. But when they start to bundle, they are very straight filaments. So the last EM that so this is very straight filament. So initially there may be just 2 or 3 of them which might be very flexible, but then later they bundle together and then they become very stiff. So that might to do with the number of filaments that are there before they can curve. Parallel, anti-parallel, that might not be dependent on condition, that might be something intrinsic to the protein. Right, yeah. Yeah, exactly. That, yes, why some form parallel. So that is what it is. That I feel that there may be residues that will come together and they can stick together only when they are in that orientation. Right. Yes, that is this filaments. So just as the filaments have come as the anti-parallel, then there is a gap in between them. So that forms a tube-like thing. So it's again just the Self-organization, exactly. There is no, and it seems like the PAR-R, the other protein, is important for that. But how PAR-R is influencing it to form this structure, or whether it is stabilizing these filaments, that is something which we don't know. Which again, we need to see in vitro and make some mutants and study them. How this tubule structure is influenced by the PAR-R. What PAR-R is actually doing to the protein is something that we need to study by structure.
So if there are no more questions, let's thank the speaker again.
Thank you. Thank you.